Every single day, millions of people open their refrigerators, pull out yesterday's leftovers, and toss them straight into the microwave without a second thought. It feels harmless. It feels normal. In fact, it has become so deeply embedded in our daily routines that questioning it seems almost ridiculous. But what if that one habit, something you have done hundreds, maybe thousands of times, has been quietly exposing your body to compounds that medical researchers have directly linked to cellular damage, hormonal disruption, and in repeated cases, the development of cancer? What if the danger is not in the food itself, but in what happens to it the moment you apply heat for the second time? This is not about scare tactics. This is not about eliminating foods from your diet. This is about understanding the chemistry that takes place inside your food when it is reheated, and why certain foods undergo transformations that your body was never designed to handle safely. Today, we are going through 4 specific foods that doctors, oncologists, food scientists, and nutritional biochemists have repeatedly flagged as dangerous when reheated. Not occasionally dangerous, not theoretically dangerous, but dangerous in ways that are now supported by peer-reviewed research, repeated clinical observation, and a growing body of molecular evidence. By the time you finish watching this, you will never look at your leftovers the same way again. Before we get into the foods themselves, you need to understand one foundational concept, because without it, none of what follows will make complete sense. Every food is a complex matrix of proteins, fats, carbohydrates, and micronutrients, all of which exist in a precise structural form when the food is freshly cooked. Heat disrupts those structures. That disruption, in and of itself, is what cooking is. The problem arises when heat is applied again after those structures have already been broken down, cooled, and in many cases reorganized into entirely new compounds. Some of those new compounds are benign, others are not. A small number of them are potently carcinogenic, meaning they have the demonstrated ability to interfere with DNA replication, trigger oxidative stress, disrupt the endocrine system, or directly damage the lining of the digestive tract. The 4 foods we are about to discuss are the ones where that second heat transformation is most chemically significant, most dangerous, and most poorly understood by the general public. The first food is rice, and this one surprises people more than almost any other on this list because rice is widely perceived as one of the most neutral, low-risk, digestively gentle foods in existence. It is bland. It is simple. It is eaten in enormous quantities across Asia, South America, and the Middle East, with very few reported problems. But the danger with rice is not in the grain itself. It is in a bacterium called Bacillus cereus. And understanding what this organism does during the cooling and reheating cycle of rice is genuinely alarming once you see the full picture. Bacillus cereus is a spore-forming bacterium that is naturally present in the soil and consequently in many cereal grains, including rice. When rice is cooked at high heat, the vegetative cells of the bacteria are killed. But, and this is the critical part, the spores are not. The spores of Bacillus cereus are extraordinarily heat resistant, and they survive standard boiling temperatures with no difficulty whatsoever. When that cooked rice is left to cool at room temperature, those surviving spores germinate. They begin multiplying rapidly. At room temperature, Bacillus cereus can double its population every 20 to 30 minutes. Within a few hours, cooked rice sitting on a countertop can contain bacterial loads that are clinically significant. Now, when you reheat that rice, you may kill the vegetative bacteria again, but the toxins they have already produced, specifically a heat-stable toxin called cerulide, remain fully intact. Cerulide is not destroyed by reheating. It cannot be boiled out, microwaved away, or neutralized by any standard kitchen process. It survives, and when ingested, it acts directly on the mitochondria of your cells. Here is where the cancer connection becomes relevant. Cerulide and related toxins produced by Bacillus cereus have been shown in multiple studies to induce mitochondrial swelling, impair oxidative phosphorylation, and generate reactive oxygen species, which are free radicals that directly damage DNA. Repeated chronic exposure to reactive oxygen species is one of the most well-documented upstream triggers of carcinogenesis. This is not a one meal causes cancer scenario. But if you are someone who cooks rice in bulk, stores it at room temperature even briefly, and routinely reheats it over months and years, the cumulative cellular stress being placed on your gastrointestinal tract is not trivial. The only safe way to store cooked rice is to cool it rapidly within 1 hour, transfer it to a sealed container, refrigerate it immediately, and consume it within 24 hours. When you do reheat it, ensure it reaches an internal temperature above 74 degrees Celsius throughout. And if rice has been sitting out at room temperature for more than 2 hours, the correct decision from a food safety standpoint is to discard it entirely, regardless of how it looks or smells. The toxin is invisible. The bacteria are invisible. The damage accumulates silently. The second food is chicken, and this one has a biochemical profile that is both well understood and widely ignored. Chicken is one of the most protein-dense foods commonly consumed, and protein behavior under reheating is where the danger lies. When chicken is cooked for the first time, its proteins, particularly myosin and actin in the muscle fiber, undergo denaturation. The proteins unfold, restructure, and set into the texture we recognize as cooked meat. That process is complete and relatively stable. What is not stable is what happens when that protein matrix is reheated, particularly if reheating is uneven, which microwave reheating almost always is. During reheating, two specific classes of compounds form that are of major concern to oncologists. The first are heterocyclic amines, commonly abbreviated as HCAs. Heterocyclic amines form when amino acids, the building blocks of protein, react with creatine and sugars at elevated temperatures. This reaction occurs most aggressively during initial high-heat cooking like grilling or frying, but it continues and accelerates during reheating, particularly in the portions of the chicken that reach higher temperatures. HCAs are classified as probable human carcinogens by the International Agency for Research on Cancer. In animal studies, they have been shown to cause tumors in the mammary gland, colon, liver, skin, lung, and prostate. In human epidemiological studies, high consumption of well-done or repeatedly heated meat has been associated with elevated risk of colorectal, pancreatic, and breast cancer. The second class of compounds are advanced glycation end products, commonly called ages. Ages form when proteins and fats react with sugars during heating, a process called the Maillard reaction, and they accumulate in the tissue of the food during repeated heat exposure. When consumed, AGEs trigger inflammatory signaling cascades in the body, drive oxidative stress, and have been implicated in both tumor progression and the acceleration of age-related cellular damage. What makes chicken particularly problematic compared to other meats is its relatively high protein density combined with its lower fat content. Fat to some degree buffers against rapid protein oxidation during reheating, and the fact that people tend to reheat chicken multiple times across multiple days in meal prep culture. If you are going to reheat chicken, it should be done once, only once, at a consistent and adequate temperature, and it should never be allowed to partially cool and then be reheated again. The internal temperature must reach at least 75 degrees Celsius all the way through. Cold spots in reheated chicken are not just a quality issue. They are a bacterial and chemical hazard simultaneously. The third food is one that many health-conscious individuals eat in large quantities and would least expect to see on a list like this: spinach and other leafy green vegetables. Spinach is celebrated as a nutritional powerhouse. It is rich in iron, folate, magnesium, vitamin K, and a broad spectrum of antioxidants. Eaten fresh or lightly steamed and consumed immediately, spinach is genuinely one of the most beneficial foods you can put in your body. The problem emerges entirely from its nitrate content and what happens to that nitrate during cooling and reheating. Spinach is one of the highest dietary sources of naturally occurring nitrates among commonly consumed vegetables. Nitrates themselves are not inherently dangerous. Your body actually uses nitrates from vegetables in positive ways, converting them to nitric oxide, which supports cardiovascular health and blood pressure regulation. The danger pathway is entirely different and activates only under specific temperature and bacterial conditions. When cooked spinach is left to cool, especially at room temperature, bacteria that are naturally present on the leaf surface begin converting nitrates into nitrites. Nitrites are substantially more reactive than nitrates. When you then reheat that spinach, particularly if you reheat it repeatedly or at uneven temperatures, the nitrites react with the amino acids present in the spinach tissue to form a class of compounds called nitrosamines. Nitrosamines are among the most potent carcinogenic compounds identified in the food and environmental science literature. The International Agency for Research on Cancer classifies several nitrosamines as Group 1 carcinogens, meaning there is sufficient evidence of human carcinogenicity. Their primary mechanism of damage is alkylation of DNA. They chemically modify the structure of your DNA in ways that directly interfere with normal cellular replication and dramatically increase the probability of mutations that lead to cancer. The organs most consistently targeted in the research literature are the stomach, esophagus, and colorectum, which is precisely the pathway that reheated nitrosamine-laden spinach passes through. This is not a remote theoretical risk. The World Health Organization has explicitly flagged the nitrate to nitrite to nitrosamine conversion pathway in cooked vegetables as a legitimate dietary carcinogen exposure route. Germany's Federal Institute for Risk Assessment has specifically warned against warming up dishes containing spinach multiple times, and has extended that warning to related leafy greens, including celery, beets, and lettuce, for the same chemical reasons. The protective measure is straightforward. Cook only the amount of spinach you intend to eat immediately. And if you do store cooked spinach, refrigerate it within 1 hour and do not reheat it at all. Eat it cold, in a salad form if necessary, or simply discard it. The nitrosamine risk from reheated spinach is not manageable through careful temperature control. It is best avoided entirely. The fourth food is eggs, and the mechanism here is different from the previous three, but no less significant. Eggs are one of the most nutritionally complete foods in the human diet. The egg white is roughly 90% protein, primarily a protein called ovalbumin. And when an egg is cooked for the first time, that protein denatures and forms a stable structure. This is normal. This is cooking. The issue with reheating eggs lies in two separate but related chemical processes: protein oxidation and cholesterol oxidation. When egg protein is exposed to heat a second time, the already denatured protein chains undergo further structural changes that produce protein oxidation byproducts, including carbonyl compounds and cross-linked protein aggregates. These oxidized protein compounds are increasingly being studied in the context of gastrointestinal health, and the emerging evidence suggests that repeated consumption of heavily oxidized proteins generates localized inflammatory responses in the intestinal lining that over time create a microenvironment conducive to abnormal cellular growth. Far more well documented, however, is cholesterol oxidation, and this is where eggs become genuinely dangerous when reheated. The yolk of an egg contains a significant quantity of cholesterol, which in its native state is a normal and necessary lipid. But cholesterol is exquisitely sensitive to heat and oxygen. When egg yolks are cooked and then reheated, particularly in methods that expose them to high heat or extended cooking times, such as reheating scrambled eggs in a microwave or pan frying a previously boiled egg, the cholesterol undergoes oxidation to form compounds known as oxysterols, specifically a group of cholesterol oxidation products referred to in the scientific literature as COPs. Cholesterol oxidation products have been the subject of extensive carcinogenicity research over the past 3 decades. They have been shown to be cytotoxic, meaning they directly kill cells, mutagenic, atherogenic, and in repeated studies associated with the initiation and promotion of colon cancer. They act by disrupting cell membrane integrity, triggering apoptosis in intestinal epithelial cells, and activating inflammatory signaling pathways, including the NF-κB pathway that is directly implicated in tumor development. The stomach and colorectal tract are the primary sites of concern because these are the tissues that come into the highest direct contact with COPs during digestion. The practical implications are clear. Eggs should be cooked fresh and eaten immediately. Boiled eggs can be stored in their shells in the refrigerator and eaten cold without risk because the shell prevents oxygen contact with the yolk during storage, limiting oxidation. But once an egg has been cooked, shelled, and exposed to air, and especially if it has been scrambled, fried, or incorporated into a dish, reheating it is something that a growing body of scientific evidence strongly advises against. If you regularly meal prep egg dishes and reheat them throughout the week, this is a habit worth reconsidering. Now, here is what all 4 of these foods have in common, and why this matters far beyond a simple list of things to avoid. In each case, the danger is not acute. It is not the kind of danger that sends you to a hospital after one meal. It is the slow, cumulative, compounding kind of danger, the kind that builds silently over years of repeated exposure, gradually increases the mutational burden in your cells, chronically inflames your digestive tissue, and shifts the cellular environment in your body progressively toward one where abnormal growth becomes more likely. This is precisely the kind of risk that is hardest for people to take seriously, because there is no immediate consequence, no clear moment of cause and effect, no single meal you can point to and say, that is what caused this. Cancer rarely works that way. It is, in the vast majority of cases, the product of accumulated insults to your biology. Insults delivered quietly, repeatedly, and often through habits so routine that they never get questioned. The scientists and physicians raising these concerns are not arguing that reheating rice once will give you cancer. They are pointing out that certain foods, when mishandled in ways that are extraordinarily common in modern kitchens, undergo chemical transformations that generate compounds with demonstrated carcinogenic potential, and that reducing your repeated exposure to those compounds is one of the simplest, most actionable, and completely cost-free adjustments you can make to your long-term health. You do not need to stop eating any of these foods. Rice, chicken, spinach, and eggs are nutritious, affordable, and important sources of essential nutrients. What needs to change is the mindset around cooking quantity and storage. Cook what you will eat. Eat what you cook. Store what remains immediately and correctly. Reheat thoughtfully at appropriate temperatures and ideally only once. And for the foods where the reheating risk is highest, particularly spinach and eggs, consider whether eating them cold or simply not storing them at all is a more practical solution than trying to manage a chemical risk that exists at the molecular level and cannot be seen, smelled, tasted, or intuitively detected. Your kitchen is where a significant portion of your long-term health outcomes are determined. Not in doctors' offices, not in pharmacies, but in the daily decisions you make about how you handle, store, and prepare the food that becomes the building blocks of your cells. The 4 foods discussed today represent 4 of the most significant and most overlooked reheating risks in common household cooking. The knowledge exists. The research is there. What changes from this point forward is entirely up to you. If this information added value to your understanding, share this video with someone who meal preps regularly, someone who reheats leftovers every day, or someone who simply deserves to know what is happening inside their food. Subscribe if you want more content rooted in science rather than sensationalism. And leave a comment below telling us which of these 4 foods surprised you the most. Because based on the research, the answers might not be what you expected.